Good afternoon. Welcome to the hundreds of you that are attending this second session of the National Primate Research Center's 60th anniversary celebration seminar series. Infectious diseases research at the NPRCs is and has always been heavily focused on HIV/AIDS for good reason. And this was clearly evidenced in yesterday's session moderated by Dr. Trotman, which featured exceptional science in the field of HIV research using non-human primates. NHPs have, however, long been leveraged as models of various other infectious pathogens and diseases caused by them. Barclay was performing infections of rhesus macaques with MTB and studying the impact of BCG vaccination on this infection in the 1960s. Bob Cedars' recent work using IV BCG to prevent TB in non-human primates is a derivative of this 50-year-old research. NHPs have since been used also to model important viral agents like HSV and influenza, bacterial pathogens like non-tuberculous mycobacteria, strep pneumo, and Neisseria, etc., and vector-borne infections like malaria and chikungunya. These models have not only investigated the pathogenesis of infection, but also the related tissue pathology, vaccine and drug development, and of late, host-directed or immunotherapy development. Over the last 2.5 years, the NHP models of SARS-CoV-2 infection have immensely contributed to both the fundamental understanding of the pathogenesis of COVID-19, but also the development of vaccines and therapeutics in this area. This session today will highlight significant contributions of the NHP models and the NPRC program to the study of infectious diseases other than HIV/AIDS. So I welcome you all. I'm Deepak Kaushal, the director of the Southwest National Primate Research Center, and we have a star-studded cast of terrific speakers today who will act along with me as panelists at the end of this session. The three speakers today are, number one, Dr. David O'Connor, PhD, the University of Wisconsin Medical Foundation Professor of Pathology and Laboratory Medicine, the unit head of the Washington National Primate Research Center Genomic Services, and a core scientist, I apologize, at the Wisconsin National Primate Research Center, and a core scientist of that primate center, and a world-renowned leader in HIV, Zika, and now SARS-CoV-2. He will be followed by Dr. Dennis Hartigan O'Connor, and then Dr. Sean Murphy, both of whom I will introduce at the beginning of their talks. So without further ado, let's hear Dr. David O'Connor speak about his work on SARS-CoV-2 recently. The presentation is titled Pandemic Interactinomes.
Hello, my name is Dave O'Connor, and I'm a professor at the University of Wisconsin-Madison, as well as an affiliate scientist at the Wisconsin National Primate Research Center. Today, I'm going to talk about maintaining primate research readiness for emerging pathogen threats in a talk I've called Pandemic Interregnums, because I would argue that we are now in a period between the emergency phase of COVID-19 and whatever comes next. First, I'd like to start with some disclosures. My wife Shelby and I are both honorary professors at the University of Melbourne in Australia. Shelby receives consulting income from Colorado State University, and I have active research funding from Amgen and perform fee-for-service genomic genotyping for many pharmaceutical companies. And before I begin, I'd like to acknowledge the National Primate Research Centers and the Wisconsin National Primate Research Center in particular for developing me as a scientist and supporting my research. I would also like to thank the many veterinarians, animal care staff, and other NPRC support staff who make infectious disease research possible. And finally, I'd like to acknowledge the animals who have participated in research to further human health. So let's start by putting mortality in perspective by looking at infectious disease mortality in the US from 1900 to 2015. Prior to COVID-19, we've been in a golden era of infectious disease mortality for about 60 to 70 years. This is long enough for most people to not remember a time when infectious diseases were a common part of everyday life. COVID-19 takes us back to the time before antibiotics were widely available. So when we talk about living with COVID and other infectious diseases, well, we've been doing that for centuries, but there's a large gamut of possible outcomes when we're living with infectious diseases, and we certainly don't want to return to 1900. Fortunately, the types of scientific advances that reduced infectious disease mortality in the first place can be brought to bear against new diseases by developing new vaccines and therapeutics. And this is beautifully illustrated by the example of HIV, shown here with its US mortality from 1980 to 2015. You can see that it was on the rise from 1980 to 1995, but then something happened. I love this headline, to the enormous surprise of researchers, because it was a surprise. At that time, HIV was a dread disease with no treatments and dim prospects for those who found out they were infected. Those of you who were around when Magic Johnson announced his retirement from the NBA and his HIV diagnosis will likely never forget it. I certainly won't. But in this study, led by Che-Chung Tsai and colleagues at the Washington National Primate Research Center, they showed that a drug, PMPA, protected pigtail macaques from being infected with simian immunodeficiency virus, the monkey version of HIV, and even protected monkeys from becoming infected even when they were treated soon after exposure. This was in November of 1995 when this paper came out, and within about a year, human trials were begun, and the resulting prodrug, tenofovir, has been used for tens of millions of person years in the ensuing 15 years as part of combination drug regimens. And this has saved countless lives. And this is a beautiful example of how science can then point the way towards better health for everyone, which can then be implemented at scale by socio-political partners such as governments and NGOs and others. Now, it took from 1981 until 1995 for PMPA to be developed and tested. And it was really unfortunate that these 15 years led to a period where HIV was this dread disease without effective treatments. However, a small, tiny silver lining is that this allowed for a robust research enterprise to develop and be supported. At the time of the PMPA study, approximately a third of NPRC activities were dedicated to SIV research. And I'm one of many scientists who benefited from the expansion of the workforce for HIV/AIDS in the late 1990s and into the early 2000s. Many of the people who are now working on emerging diseases like SARS-CoV-2 spent years studying the immunology, virology, and pathogenesis of HIV and SIV. And indeed, the robust support for SIV research at the National Primate Research Centers has created a collaborative workforce of junior, mid-level, and senior scientists, a training pipeline for researchers, veterinarians, and clinicians that span the complete translational research spectrum from bench to animals to bedside and beyond. And that's typically what we think about when we talk about translational research, but I think there's another definition that we should use as well. This map shows new human diseases from 1981, beginning with HIV, through 2020. And I really want to highlight that the risk of new diseases is ever present. This means that we need to have heightened and sustained readiness to respond quickly whenever there is a new threat. And the sort of translational research that was initially supported by SIV can mean that we can bring knowledge and expertise from one emerging disease to another, indeed translating the knowledge gained in one disease to study another. And we saw this vividly in the movement of research knowledge, first from SIV, HIV, and AIDS, to the Zika virus pandemic in 2016, to COVID, to whatever is going to come next. And so let's talk about what some of these knowledge translations looked like. First, I want to start with Zika virus. So I was working on SIV from 1997 to 2015. And as part of that, I was very interested in the evolution of the virus. We developed deep sequencing techniques to study immune escape. Those same techniques could be used to study drug resistance. We wanted to make this available to researchers in low and medium income countries. And so we were teaching researchers in Brazil how to do deep sequencing. Here I'm showing a picture with Renato Santana, a collaborator of mine from Rio de Janeiro, with my wife Shelby and our son Eli, in Florianopolis, Brazil in October 2015. Later that day, Renato told me that in Recife, in the northeast of the country, there were babies being born with small heads, and there was a suspicion that a virus was involved. We began assembling the permissions necessary to look at some of the people who were infected or who were affected to see if they were indeed infected with the virus. And by the time we had those permissions in place, other groups had shown that there was a correlation between infection with the Zika virus and babies being born with small heads, this condition called microcephaly. Zika virus is a classical neglected tropical disease. It was first discovered in 1947 in Africa, and very little work was done on it in the ensuing decades. So when we started working on Zika virus and trying to understand this new epidemic of microcephaly, we had to all start from scratch. Enabling this, we began a very open and transparent collaborative research process that involved researchers from around the world. Everyone was welcome as long as they agreed to be collaborative and share information with one another. Here you see Sally Permar, now of Cornell, Ted Golas from the University of Wisconsin, and me brainstorming early experiments in Slack in December 2015. The use of these sorts of collaboration tools is one of the technological advances that accelerated discovery with Zika virus, and it's a theme I'm going to come back to later in the talk. By February of 2016, when cases in Brazil were surging, we received approval to infect macaques with Zika virus. This is Tom Friedrich with the cooler that contained the challenge virus that we used in these first animals. And a few days before this challenge, Tom and I made a fateful decision. We recognized we were going to be among the first to have macaque data with Zika virus. And so we made the decision to make this data available to everyone. We were already beginning to maintain a portal so that we could share our data with all of our collaborators. And due to the way the portal was set up, we could flip a switch and make it available to the entire world. And indeed, that's what we decided to do. So when we launched this portal in mid-February 2016, it looked like this. We talked about what we were doing, and then we had data from our study being put online in real time. We were worried, of course, about what the implications of this would be. Would animal rights activists seize upon this? Would people complain about this very different way of sharing scientific data? The answer was no, not really. In fact, we had the opposite reaction. Other scientists began to contribute and suggest ways that we might improve our experiments. And through all of this, we were able to show that macaques could be infected with Zika virus and that these animals would develop immune responses that protected from reinfection with both Brazilian type Asian lineage Zika virus, as well as more divergent African lineage Zika virus. Over the last 5 years, we've extended this work to study congenital Zika in infant macaques using technologies that were originally developed to study fetal alcohol syndrome in macaques. We've found Zika virus frequently in maternal and fetal tissues, as well as at the maternal-fetal interface, and we've discovered that a hyperimmune globulin treatment administered soon after infection can arrest virus replication. And most recently, we found that African lineage Zika virus, when given to pregnant monkeys at high doses and early in infection, can cause a surprisingly high number of miscarriages. This makes us wonder whether or not Zika might be having an impact in Africa where Zika-associated miscarriages could be missed due to other causes. So some of the key lessons that we've learned here are that frictionless internal and public discussions are essential, that real-time data sharing accelerates science, improving rigor and reproducibility, and that big tent collaborations can bring different types of expertise to bear on a complex and challenging problem. We also learned that the public is generally receptive to learning about how macaques are used in infectious disease research, as long as this is presented professionally and respectfully. So how did we apply these lessons when COVID emerged? We basically broke out the same playbook. We started a Slack collaboratory, which grew to have more than 150 people in it, starting in January 2020. We basically told people that they had to share their findings and ideas with us if they wanted to participate, and we were glad that many, many people did. And by April, we had worked to start a challenge study at the National Institutes of Health's integrated research facility in collaboration with their scientists. We once again put data from this available, made data from this available in real time on an open data sharing portal. We showed that these animals developed mild clinical disease and had evidence for significant inflammation and infection in their lungs. And this work, along with the collaborative work of other groups such as the Active Collaboratory, really helped propel the use of non-human primates in COVID vaccine development. So much so that by early 2021, this received international attention. The New York Times did a story about how macaques were essential for COVID vaccine research, and that this has created a supply shortage of animals. Again, illustrating the importance of sustainability in a response. So what may happen next? What have we learned from the 5 years after the Zika virus pandemic that might tell us something about what's going to happen with COVID? Well, first, I think we have to ask questions about sustainability. In my mind, there are two different ways the pandemic could go from here. The first is that, like Zika, we have an era where the chaos of an active pandemic gives way to a quieting down. The vaccines that we have, the monoclonal antibodies, the treatments, all mean that the worst of COVID is behind us. It's also possible that COVID's going to take a trajectory more like HIV, where we're really only in 1983 or 1984 in a pandemic who's going to be measured out over decades. And I think an honest scientist is going to have to answer that we simply don't know what this virus has in store for us in the future. And so we have to think about what is sustainability going to look like in terms of developing a research agenda around COVID. And indeed, others are already asking this question. This is an article from The Atlantic from about a week before I'm recording this talk, talking about how the US Congress has recently reduced coronavirus funding in the United States because there are other competing priorities, such as helping to subsidize international affairs such as the Ukraine situation, as well as other priorities that may have been neglected during the 2 years during the COVID emergency. But in addition to sustainability, we have to think about what about the research and data products that were generated during COVID. So this is a picture of the Public Health Madison and Dane County data dashboard. I'm sure many of you have similar dashboards in your communities and states and countries. And these are challenging because they take time and energy to maintain and keep up to date and keep accurate. And they're very vulnerable to link rot, meaning a few years from now, are they still going to work? Are they still going to be maintained? Are they still going to have active support to keep them updated? And in fact, this is a question that we've had to wrestle with our Zika virus portal. We still maintain it. You can still go to it today and see it, but it's rarely visited and it's difficult to maintain. And that becomes ever more difficult as we try to put in ever expanding data. Who is going to be funded to sustain these resources, these dashboards, these other data sharing portals? Should they go away? What happens and what knowledge gets lost if they simply go away or suddenly one day they just become no longer accessible? This is a problem that I think we really have to think about. We also have to think about what the future will look like for junior scientists. This is a picture of Gage Moreno. He was a PhD student in my lab, and he really came into his own during the Zika virus pandemic. During the COVID pandemic. Gage is shown here receiving our first COVID sample or SARS-CoV-2 sample for deep sequencing in mid-February 2020. Over the next year and a half, he published a flurry of papers, defended his PhD, and went to Pardis Sabeti's lab at the Broad Institute to do his postdoc. Well, what is Gage, who has extraordinary talent studying COVID, going to do if COVID research funding dries up? If we don't have the stability of a long-term research agenda like we did for HIV, how will the talent of this generation of scientists be harnessed? And what about the next generation of scientists? We have kids who are as young as kindergarten, who are in high school and college, who have lived through this pandemic, and instead of becoming investment bankers or working for tech firms, might want to come and contribute their enormous skills to virology research and are considering a career in science. Will there be career opportunities for them? Will there be a deep bench for future pandemics? And I think we have to think about all these things because there will be new emerging pathogen threats in the coming years. And maintaining the researchers, facilities, and research projects will be important to have an agile response when this occurs, whether it's with a continuation of SARS-CoV-2 and COVID, or whether it's something else. This also requires investments in infrastructure, computational resources, animal availability, genomics and other omics resources so that we can learn more from each individual study, high containment laboratories, so that we can make science responsive to SARS-CoV-3 or whatever other infectious disease threat comes next. Because I think no matter what comes next, non-human primate studies are always going to be essential when new threats emerge because they are so similar to humans and they so often predict what's going to happen with human infections. And in fact, in March of 2020, some of the earliest non-human primate data showed that animals had very high viral loads before there were clinical signs. And that led to this idea that asymptomatic transmission might be very common with SARS-CoV-2. This was some of the earliest data that drove the use of face masks and drove the use of risk mitigation strategies that focused on protecting people from infection because they might be around someone who is asymptomatic but was nonetheless highly contagious. Again, this was a lesson that we first learned with HIV about 4 decades before. And so I just want to close by once again reiterating that the NPRC system has been essential to past pandemics such as HIV/AIDS, present pandemics such as COVID, and it will be essential to fighting future pandemics that will emerge during the next 60 years. Congratulations to everyone who's involved in the 60th anniversary of the NPRC system, and congratulations to all who are participating in this celebratory conference series. Thank you very much.
And congratulations to you, David, for the wonderful work you and your group have done in this arena. I'm sure there will be questions because of this thought-provoking and exceptional talk that David gave, that Dr. O'Connor gave. But all attendees in VOVA, please post your questions in that app, and the panelists will receive them, and we will tackle these questions at the end of the 3 talks in the Q&A session, which should happen at 3:35 Central Time. In the meanwhile, we'll move on to the second talk to be given by Dr. Dennis Hartigan O'Connor, no relation according to my book. And this is titled Targeting B and T cell interactions for Durable Immune Responses to SARS-CoV-2. Dr. Hartigan O'Connor is an associate professor and a core scientist of the California National Primate Research Center. Thank you.
My name is Dennis Hartigan O'Connor. I'm an associate professor at the University of California, Davis in the Department of Medical Microbiology and Immunology, and a core scientist at the California National Primate Research Center. I'll tell you today about our efforts to target B-cell, T-cell interactions as a key mechanism for inducing durable immune responses to SARS-CoV-2. And this work really began in the earliest phases of the pandemic when it was still a question whether we could induce meaningful immunity to SARS-CoV-2. And like so many of us coming from the HIV field, I think that was a real unknown. Our big question was whether a vaccine would be possible and what that vaccine would have to do. Our first work at the Primate Center with SARS-CoV-2 was focused on testing convalescent plasma as an intervention in the disease. And honestly, those results were a little bit discouraging and suggested that it might be very difficult to protect against SARS-CoV-2 using antibodies, which it later turned out, of course, you can. Our first attempts were with relatively low titer plasma, which had an NT50 of 1149. That was infused just a day after infections as sort of the optimal timing that you could imagine for successful intervention. And the results were minimal in terms of virologic or clinical impact. And later, a second attempt was made, again, as a large collaborative group, investigators at CNPRC using a higher titer plasma, also infused a day after infection. And here again, we saw no antiviral effects, although there were some lessening of the pathologic process. The results of those early experiments made me feel that it might be very difficult to protect against this disease with vaccines that produced only a low titer of antibodies. The first signs of hope for me came from the earliest vaccine studies of Dan Baruch and a number of groups. But I remember particularly this figure as one that struck me as very hopeful, which was showing from across a range of macaques that received DNA vaccines, what titers seemed to be completely protective against subgenomic mRNA in the lung and those that were only partially protective. I think the mean titer here is about 1 to 300 of neutralizing antibodies. This seemed to be the first time I began to think about a numeric goal that could be achieved and that would provide protection to most people. That's a bit of a way star as we think about boosters and maintaining protection in people who are vulnerable is what is the titer at which we need to pay attention and consider the need for boosting vaccination. And we still don't know that answer, and there's not one answer for everybody. That question of longitudinal protection and durability began to be explored quite early in the race to produce a vaccine. These are data from, I believe, the first phase 1 clinical trial of Moderna, published by Alicia Widge in the New England Journal of Medicine, showing that already only 4 months after vaccination, a decline in neutralizing antibodies was apparent that might be enough to be clinically relevant. Here, for example, most younger people exhibited relative stability, but a couple saw declines in their antibody titers that would be expected based on those early ideas about what titers might be protective in macaques that I showed on the previous slide to be problematic. And in older groups of individuals, you can see a much more consistent decline across individuals and a larger fraction of people who might lose antibodies to the point that you would imagine protection would be incomplete at best. These early data were later corroborated by larger studies with a variety of different vaccine types. The key figures here on the right, which is that, again, even within the first 4 months, you see a meaningful decline that brings antibody titers to within striking distance of that possible threshold we talked about of a few hundred. Of course, these findings overlapped with the emergence of the first variants. For the whole world, really, durability of vaccine-mediated protection became a primary concern. And indeed, remains a concern today as we search for a vaccine that might be durable over the course of many years or decades. Our thinking about this in my laboratory focused on the germinal centers where B cells and T cells interact, in part because we knew very little about B cells at the beginning of the SARS-CoV-2 pandemic. Our primary work is in T cell vaccines for HIV. And so we were very interested in what contribution T cells make to durable immune response. The cartoons here show those interactions between B cells and T cells that first recruit B cells into the germinal centers and then guide their development either to plasma cells that secrete antibodies, memory cells that seem to serve as a source of diversity in future immune responses, potentially to booster vaccines, and those that drive somatic hypermutation, evolution, and increased affinity in the dark zone of the germinal center. It was our hypothesis that by forcing this interaction to occur, we might be able to evade a number of these checkpoints on B cell development. The idea is to remove this competitive limit to the number of B cells that develop. The approach that we took is one that we call a bidirectional targeting, which is the linkage of the immunogen, in this case, the receptor binding domain of SARS-CoV-2, to an anti-T cell, a single chain variable fragment. And in this example, we're showing an anti-CD3 scFv because that's the one we've done the most work with. But conceptually, it could be really any anti-T cell fragment, and specifically one that interacts with T follicular helper cells and that bridges the cell on the left of the screen, the B cell, with the cell on the right. And it does that because, of course, the cell on the left is an antigen-specific B cell, which binds to the left half of this fusion protein, the RBD half, the immunogen, and the cell on the right of the screen, the T cell, bears those surface receptors that the scFv fragment is susceptible to. So this is essentially immunotargeting for T cells, and this is the approach we've been testing in non-human primates as a way to induce durable immune responses. The idea is to provide indiscriminate help to amplify antibody production and plasma cell generation, really at the simplest level by increasing the number of naive B cells that are recruited. We think that these bidirectionally targeted immunogens might be working via a number of different pathways, but the one that we envisioned is shown here. The normal process of B cell development in the lymph node involves a competition between B cells for and a development only of those B cells that are able to acquire enough to internalize the antigen, process it, present it on MHC class II molecules, and then interact with T cells. And the idea is that the production of this immunogen might reduce the competition for antigen internalization by allowing productive interactions and T cell signaling to B cells without necessarily the internalization and processing of all this antigen. The results show, we think, that there's a number of different mechanisms at work, and these are a few that you might think about as we go through the data. The mechanism shown on the left here is really the one that inspired the work, in which the arrayed antigen on the surface of antigen-specific B cells sends a non-specific signal to T cells that allows them to produce the help that's needed for B cell development. But we think there's some evidence that other things are happening as well. We see an expansion of antigen-specific T cells, and this could be mediated by increased uptake and processing of antigen by those activated B cells undergoing interaction, providing a greater quantity of MHC2 peptide complexes on the surface that can then signal to antigen-specific T cells that might be in the vicinity. And then the duration of B cell T cell interaction seems to be an important rheostat that controls B cell development, and possibly that could be a contributing mechanism as well, although it's not one we've started to explore yet. And this idea extends to a number of different T cell targets. As I mentioned, we've started to explore a number of them. Here I'm just illustrating the anti-CD2, anti-CD3, and anti-CD4 approaches that we would call, for short, S2, S3, or S4, that might have different properties and that might confer different properties on the antibody response. So the potential of S3 is obviously to activate T cells when the immunogen is arrayed on the surface of B cells, and that may contribute to the efficacy of the immunogen, or it may not be important. And an immunogen like S4, RBD, for example, would potentially lengthen those B cell T cell interactions or provide proximity between the T cell and the B cell, but without any potential for activation. And anti-CD2 linked immunogens might drive interaction with a different set of cells. And so I'll tell you about S3, but we're pursuing all these approaches. The first thing we did was to evaluate the binding characteristics of these antibodies, and we were able to develop candidates for all of them that do bind as predicted to the cell subsets that bear those receptors. And in particular, I'll just draw your attention to S3-RBD in the middle of the second row. This immunogen is the one diagrammed above, in which the anti-CD3 fragment is linked to RBD. And what's shown here on the y-axis is CD8, and on the x-axis is RBD. And so what's on the x-axis is binding of an additional secondary anti-RBD antibody to labeled cells. And what's shown is that both CD8 positive and CD8 negative cells, which are presumably CD4 positive T cells, can bind to this S3-RBD immunogen. And you can sort of imagine this immunogen arrayed on the surface of these cells. And the first experiments that we did were really simple DNA and Ad35 vaccination experiments, in which the immunogen in the priming step was either RBD alone or S3-RBD. 4 weeks later, the animals are boosted with an Ad35 expressing RBD alone. So we're really focused initially on that idea of increased B cell recruitment, of democratized recruitment of naive B cells. And the readouts I'll tell you about are binding and neutralizing antibodies, circulating memory B cells, and TF8 cells. One question I like to address at the beginning when we're talking about anti-CD3 is safety. These immunogens are monomeric, so it's a single anti-CD3 scFv fragment linked to RBD. And we find that this monomeric immunogen does not induce any significant T cell activation in vitro. You would see that here in these These are just duplicates. Whether you add S3-RBD into the medium or you don't, you see a similar negligible level of cytokine production in T cells and PBMC. But when you bind this monomeric immunogen to a plate, then you do start to see cytokine production. And of course, in my mind, the hypothesis is that this binding to the plate and production of this immunogen array mimics the binding of the immunogen to a B cell and its presentation to the T cell in an array. And we also find that when we immunize animals with vaccines that either make RBD or S3-RBD, there's no difference in the clinical condition of the animals or in body temperature and cytokine production or anything else. So we don't really see evidence for a mechanism by which this might be stimulatory in an inappropriate setting, and we don't see any clinical evidence of toxicity. We've done experiments sort of in two age groups, approximately 1-year-old animals and much older animals. We see a similar pattern of binding antibodies in both. So on the left, what we are looking at are the endpoint titers in animals that received either S3-RBD priming, and those are in red, or RBD alone priming, and those are in green. And we do see an increase in the peak antibody titers, which is something that we had hoped for, and maybe we'd hoped for a bigger difference. But the really dramatic difference between the two immunogens comes over the span of the next 6 months, which is this critical period in which we're thinking about human antibody declines after receiving vaccination. And here the difference between the new immunogens expresses itself, and we see a much greater stability to the binding antibodies that are being produced after priming with S3-RBD, such that after 6 months, the animals that received S3-RBD have a 10-fold higher antibody titer. And in the older animals, we see a similar pattern. We are really focused as a future application on older individuals, immunocompromised people who might benefit most from this kind of strategy. And we see a similar pattern in which those peak antibody titers are higher after receipt of the S3-RBD, and there's also greater durability to the response, within about a 10-fold difference after 5 months in this case. The differences in terms of neutralizing antibodies are more impressive, and I'm not sure if that reflects the presence of more antibodies or just the sort of higher functional bar that antibodies have to meet, such that as those titers start to decline, the neutralization titers are on a bit more of a cliff's edge. But the difference that we see in neutralization is that recipients of RBD alone, again shown here in green, have declines such that the neutralization becomes really undetectable in this experimental model by 6 months in many animals. So here it's 2 out of 3, but it's not at all unusual for us to see declines that are more precipitous in the neutralizing antibodies than in the binding antibodies. And in contrast, the neutralizing antibodies in recipients of S3-RBD show the same stability that the binding antibodies showed, and we think reflects the continued presence of an adequate quantity of antibody. And in absolute terms, though, it's very difficult, obviously, to compare across pseudovirus assays. The titers that we achieve after, in this case, 8 months of 1 to 1,000, we think are likely to be protective, and I think compare favorably to what's seen with the clinical vaccines that are used today. We also looked at circulating IgA titers because we're interested in the idea that immunogens like this might drive maturation of the B cells in such a way that they produce more immunoglobulin A that might be protective at mucosal surfaces. And we do see more IgA in recipients of S3-RBD than of RBD, although I don't think we've been able to show that the IgA is a bigger fraction of what's produced. So these greater quantity of IgA may just reflect the greater quantity of antibody. And so we don't know yet if these bidirectionally targeted immunogens have the capacity to preferentially induce IgA. And we also do find a higher quantity of circulating memory B cells. Again, these data are just from the younger group of animals. And what we're looking at on the left are IgD negative, CD20 high memory B cells stained with 2 RBD tetramers, so that those cells at the upper right are memory B cells induced by the vaccine. And we also see a larger quantity of these cells that, again, seems to be relatively stable during the first 4 months after vaccination. We've only just started to look at the specific antibodies that are induced. We've cloned about 100 antibodies at this point, although we've only evaluated 7 for both binding and neutralization. What we found is that of those 7, we have 4 very good antibodies with high affinity in the single nanogram per ml range, which compares favorably to antibody evolution in people recovering from COVID-19. So on the right-hand side, we see our 4 binding antibodies compared in terms of EC50 to evolution of antibodies in COVID convalescents. But we don't know as a population whether there's going to be any difference in the extent of affinity maturation or the affinity that's seen with these antibodies. Finally, the most surprising outcome maybe for me, because the strategy was focused on non-specific T cell activation, was an enhanced frequency of T follicular helper cells that are specific for RBD. So the left part of the screen here shows our activation-induced marker assay in which circulating T cells are exposed to peptides, and then those T cells that have the surface characteristics of TFH are monitored for surface markers that indicate activation. And we compare control stimulated cells, which are labeled DMSO here, to those stimulated with RBD peptides. And we monitor a number of different markers. 2 shown here are CD25 and CD137. And in the examples on the left, you can see that there's an expression of more CD25 and CD137 on stimulation of animals that received S3-RBD than RBD alone. Again, this is just in the younger age group, but we do see similar results in the older age group. And on the right, you can see that the circulating TFH cells are stable again for about 5 months after vaccination. So again, this wasn't a predicted result for me, and the mechanism that I've proposed as a possible explanation, the stimulation of better antigen processing and presentation by those B cells that are brought into proximity with T cells. So finally, we've taken the same regimen and transferred it into a complete adenovector regimen with Ad26 priming and Ad35 RBD boosting. The basic experiment is the same here, in which priming is with Ad26 RBD alone or Ad26 S3 RBD, followed by boosting 4 weeks later by Ad35 RBD. And we see on the right a very similar pattern in immune responses. Here again, the S3 RBD recipients are in red. In this case, we see a peak immune response that is indistinguishable between the 2 groups. So we didn't get that boost in the peak antibody response that we had previously seen, but we do see, again, this tendency to a maintenance of neutralizing antibodies over a longer period of time in S3 RBD recipients that we think is a desirable feature of future COVID boosters that might be administered less frequently than every 6 months. So in summary, short-term protection and emergence of resistant variants are synergistic threats to vaccine efficacy that are playing out today, as we seem to need boosters, in my opinion, far too frequently. And I think we'd all like to get to the point where we might be able to achieve broad protection and high titers that are persistent over a long period of time. S3 is an adjuvant-like tool. We've had people complain about calling it an adjuvant that's designed, in any case, to overcome pre-existing dominant responses by providing indiscriminate B cell help and recruiting clones that might not otherwise be recruited. This idea of recruiting subdominant B cells is something that we haven't begun to address. What we've seen in our experiments so far is that priming vaccination with S3-RBD provided durable neutralizing antibody responses, as well as more memory B cells and more antigen-specific T follicular helper cells. And we propose that this might be an ideal boosting vaccination strategy that would allow for high titer boosted antibodies that are durable for years rather than months, that could provide durable cross-variant protection and robust IgA responses as well. So this work has been really driven in my laboratory by William Chang and Joe Dutra. William is a true B-cell immunologist who was trained with Nicole Baumgart. Joe is an extraordinarily talented molecular biologist who made all of our vectors and constructs. But really the whole lab was involved in these experiments that took such a huge effort at the beginning of the pandemic. Jesse Deere in particular is the virologist who undertook to enter the BSL-3 and do all the work with SARS-CoV-2 at a time when really the danger of working with SARS-CoV-2 was unquantifiable, and courageously stepped in and did all that work. And then finally, our funders, I want to mention that the California National Primate Research Center made an extraordinary coordinated effort to address the pandemic, and it was inspiring to work together with my colleagues to get these experiments done. NIAID is our primary source of funding, and then the University of California provided really critical seed funding to allow us to get started with evaluating these strategies.
Thank you, Dr. Artigan O'Connor, for that insightful talk. And in fact, if I may draw from your acknowledgements, and tell everybody, and many of you attending probably know this, that the entire NPRC consortium, all of the 7 primate centers, stepped forward at the time that the pandemic was early breaking and provided out-of-turn pilot grants to jumpstart many projects in many of these primate centers. And if I may, also, the directors were guided in doing so by Sherry Hill, the program officer at the NIH. I know many of the NIH folks are on today. So credit is due to her for her visionary guidance to all the directors at that point of time. I know Sherry Hill is going to retire very soon, and we must acknowledge her help in this direction. The third talk today will be delivered by Dr. Sean Murphy, Associate Professor of Laboratory Medicine at the Washington National Primate Research Center and the University of Washington. He studies the immune response to complex infectious diseases, focused on diagnosis and primarily focused on malaria. And the title of his talk is The Hunt for Vaccines and Diagnostics to Accelerate Malaria Elimination and Eradication.
Hello, everyone. My name is Sean Murphy. I'm an associate professor in the Department of Lab Medicine and Pathology at the University of Washington. And I would like to thank the organizers for inviting me to be a part of this 60th anniversary celebration. So I'm going to tell you about the work we do in our group to try to develop a highly effective malaria vaccine, and in particular, how we leverage non-human primate resources to accelerate this process. I have a couple of disclosures. We get research support from a number of startup companies that work to develop malaria vaccines or other kinds of vaccines, Scenaria, Malorvax, and Lumen. And then I also have equity in a company called Sound Vaccines, which is trying to commercialize DNA vaccines, though at the moment, none of them are related to malaria. We know a lot about malaria. We hear about it in the news, and yet, to many Americans, people think that malaria is something that has gone away. And for many decades, malaria control was, in fact, headed in the right direction. However, since about 2015, progress toward malaria elimination or at least reductions has stalled. And in some parts of the world, those trends have reversed, particularly in the highest burden countries in Africa. This is a graph showing that reversal of fortune from the 2018 World Malaria Report. And if you look to the most recent World Malaria Report, the World Health Organization estimates that 241 million cases lead to about 627,000 deaths in 2020, a year that was made obviously worse by COVID-19. If we look at the malaria life cycle, after a mosquito bites a person, parasites are transmitted to the liver, and they live there for about a week before the parasites go into the blood, where they reproduce every 2 days in the case of Plasmodium falciparum, and in the blood, they cause all clinical disease. At some point in the blood, they go on to form gametocytes, which are not disease-causing in humans, but propagate the life cycle back into the mosquito. When we think about making a vaccine against the parasite, this slide highlights all the different places in the life cycle where different parts of the immune system could provide some protection against infection. And in our case, we are most focused on the liver stage because we can leverage the widest array of immune components, in particular CD8 T cells, to try to kill all of the infected liver cells before they go on to cause the disease-causing blood stage. And so at the end of this talk, I hope you understand why CD8 T cells are so critical to our storyline here. Recently, the World Health Organization also approved a vaccine that you may have heard of called RTSS. But the reason this isn't the end of the story for malaria is that RTSS, while important, has relatively low efficacy. And it is not a vaccine that will precipitate the end of malaria. The liver stage is an ideal target to make the malaria vaccine because if you can stop all the parasites in the liver stage, you can prevent infection from progressing to the blood stage. The problem is that even one infected hepatocyte, which is what's depicted in this picture, is capable of making 30 or 50,000 progeny cells in just 7 days and even one of those infected hepatocytes, when it bursts, is able to seed the bloodstream and start the blood stage infection. So we need 100% protection in the liver, not 99%. For the past 40 plus years, one of the most potent tools in the malaria vaccine arsenal has been the use of live attenuated sporozoites. This is the form of the parasite that comes out of the mosquito, and several flavors of sporozoite-based vaccines have since arisen. You can give wild-type sporozoites completely unattenuated, and they will go through the liver, and if you at the same time give someone chloroquine to kill the parasites as they emerge, you have a broad range of proteins expressed throughout the liver stage. You can also irradiate to make radiation attenuated sporozoites, or you can start deleting genes and make genetically attenuated sporozoites. And you can design these to stop at different times and different stages of progression in the liver. The reason sporozoites are important is that they actually are amongst the most highly protective experimental malaria vaccines that have been developed so far. And the reason we know this is because we have tested them, we and others have tested them in controlled human malaria infection trials that followed on the heels of course, mouse and non-human primate studies over the past decades. If you take irradiated sporozoites, we know that we can achieve very high rates of protection against homologous challenge and lower, but still impressive rates of protection against heterologous challenge. This is where the sporozoites were derived from one strain of Plasmodium falciparum and the challenge strain, a different strain. And so you can see here that we can, in these controlled human infection studies, achieve rates, well above 50%, and in some cases near 100%. If we look at people who go through controlled human malaria infection studies having been vaccinated with sporozoites, there is one study that I think has a remarkable finding that implicates the role of CD8 T cells. And that is from Andrew Shizuka at the VRC at the NIH. They took a group of people who were vaccinated with sporozoites according to this schedule. The details don't really matter. And then they were challenged several times. One group was challenged about 10 weeks from vaccination, and then not again until the 80th week of the study, effectively almost a year after their last encounter. And these people were protected against challenge. And when you look in their sera, you cannot find antibodies that explain why they were protected. And you cannot really find T cells either. And so what is responsible for the protection? And the answer is T cells, but they're not in the peripheral blood, they're in the liver. And these are CD8 positive liver resident memory T cells, depicted here in a beautiful video from Bill Heath's group in Australia with a TCR transgenic version that can be tracked. And these are T cells that reside in liver sinusoids, and they recognize class 1 MHC on infected cells and express the markers shown here. But to get these to be formed, you need to express the cognate antigen that they target in the liver. So in our group, over the last 5 years now, we've been working on a concept we call prime and trap vaccination, intended to induce liver resident memory T cells. Since the liver is not a great place to prime an immune response, we instead prime with a DNA vaccine, or in some cases, more recently, mRNA vaccines in the skin. And then in the first generation of prime and trap vaccine, we come and give sporozoites intravenously, which are professionals at homing to the liver. And when we do this, the prime cells in the skin, some of them traffic to the liver to form resident memory T cells. We know that if we do this, DNA prime and irradiated sporozoite trap, we can protect almost all the mice we have at essentially a long-term time point. And we know that this protection hinges on the presence of these resident memory CD8 T cells. So we have many lines of work in the lab trying to bolster this finding, to refine the vaccine, and to move this closer to the clinic. Non-human primates play an indispensable role in the evaluation of this prime and trap vaccine, and really for any malaria vaccine, but especially those like ours that depend on tissue-specific CD8 T cells. The work I'm going to show you that highlights our non-human primate effort is led by Dr. Melanie Shears in our group, who is an acting instructor in our department and who is really an expert on non-human primate malaria. So the first takeaway from non-human primates and why we need them in our research is that they really help to define the critical number of liver-resident memory T cells that are required to protect an animal. Our goal is to do dose de-escalation studies with the vaccine and get to a point where we have a dose that protects and a lower dose that doesn't protect. And then by sampling the liver and doing flow cytometry, we hope to be able to define how many T cells are needed per cubic block of liver. And we can do this with mice and we can do this with non-human primates, but we of course cannot do this with humans. And so because non-human primates are a large animal model, this accessibility of the liver is of critical importance. The second takeaway is that in order to get that liver, we needed to develop safe surgical techniques, and we did so with the skilled assistance of our Washington National Primate Research Center veterinary surgeons and staff. So we developed a liver wedge resection method that we applied in this vaccination and then re-exposure model, where after we vaccinated, but before we re-exposed the animals, we obtained a relatively large liver biopsy and did so successfully in 9 out of 9 non-human primates without any complications. And we found that we got the highest levels of T cell responses in those liver lymphocytes when we used a prime and trap vaccine model shown here with the ELISpot data for interferon gamma. And we're now using this in additional vaccination challenge studies that are coming up. Third, curiously, in mice, the liver stage completes in just 2 days. However, in Plasmodium knowlesi in monkeys and Plasmodium falciparum in humans, the liver stage takes about 6 days. This difference is hugely consequential and very much overlooked by our field, because in a mouse, after 2 days, the only thing that will be different than it was on day 0 is that the T cells that were there can become activated, but they will not significantly expand. In contrast, with 6 days, we have a much greater ability to have the immune system show its full might, because in the first couple of days, yes, those T cells will become activated, but on days 2 through 5 or 6, they will also expand, and the contribution of both expanding and trafficking T cells will result in more T cells being present in the liver and potentially greater control. Fourth, we have studied both Plasmodium falciparum and Plasmodium knowlesi, and we have found that these are not equally infectious. Now, this isn't surprising, but for many years, people have attempted to inject P. falciparum sporozoites into non-human primates in the hopes of obtaining highly meaningful immunogenicity results to inform human trials. And what we found was that when we sampled the liver for parasite bioburden 6 days after injecting sporozoites, we found that while we could find the presence of some P. falciparum, the human parasite, in the non-human primate liver, it was nothing compared to the matched Plasmodium knowlesi parasite, which is the natural pathogen that infects these non-human primates in the wild. The amount of liver parasite burden, despite giving half or 10 times less of this parasite, was about at least 4 orders of magnitude higher than it is for P. falciparum. So this is very informative to us and really means that we need to use these species matched parasite host pairs going forward. Fifth, we also have come to realize that not all Plasmodium species that have been used in non-human primates are equally pathogenic in different hosts. And by this, I mean that we have used now both rhesus macaques and pig-tailed macaques and found that a parasite Plasmodium knowlesi, that causes severe disease in rhesus, causes actually pretty benign disease in pigtailed macaques. So Dr. Shears has been working on method development and model development in the pigtail model. We were forced to use the pigtails because of the reprioritization of rhesus for COVID-19 research. But very interestingly, we found very consistent times to infection. And yet, despite the fact that the time to infection is very similar to rhesus macaques, unlike rhesus, when the pigtailed macaques shown on the right here reach about 12 days into their infection, that infection plateaus. It does not go away, but the animals are largely asymptomatic and the infection just simmers along until we drug treat them and cure them. We are now using these two models to play off an acute infection in rhesus macaques and a chronic infection in pigtailed macaques to try to understand different malaria exposure scenarios that are very relevant to what people experience in the field. So in our group, non-human primate studies started out as a pilot area for our research program and have become a central pillar to our malaria vaccine development program. Over the past 5 years, they've added key insights about vaccine-mediated protection. They give us access to a liver from a host that has a duration of the liver stage that matches what we see in humans. We have models that have emerged that allow us to mimic varying degrees of previous malaria exposure, and we have leveraged these into an NIH U01 grant with OHSU. This model serves as a stringent large animal model to look at outbred immune responses. And so recently, in data I don't have time to show you, unfortunately, we've even managed to protect some pigtailed macaques using prime and trap vaccination, using DNA prime and P. knowlesi sporozoite trapping, and we look forward to publishing that data soon. And this kind of model is really needed because there are an increasing variety of malaria vaccine candidates, and we think the non-human primate models can really help us prioritize and match vaccine outcome to vaccine mechanism. Along the way, this has enabled a lot of great collaborations, great career and educational opportunities, and we hope has helped to accelerate malaria vaccine development. I have many, many thank yous. I don't have time to thank everybody on this slide. I would like to point out again and very much thank Dr. Melanie Shears, who is a real expert and wonderful collaborator here at the University of Washington. And I would also like to thank all of the staff and scientists at the Washington National Primate Research Center for bringing us into your circle and to helping us learn and advance our malaria vaccine program over the last several years. Thank you.
Thank you very much, Dr. Murphy, for that wonderful, wonderful talk. I think we will soon be ready to go into a panel discussion of myself along with our three speakers today. And I do know that there are some questions for each one of our speakers. So we will take them, but if I may take just a minute to review what we've learned today, I think all of you that are attending will agree with me that we've had wonderful sharing of data, and we learned how developing expertise during the time of Zika really enabled and springboarded some of our researchers to perform initial experiments on SARS-CoV-2, essentially the word playbook. We must keep the playbook ready at all time, and we must have our quarterbacks, our running backs, and our receivers be conversant with that playbook at all periods of time. And then we learned that wonderful immunology, fundamental immunology work has been done, vaccine immunology work has been done. In just a couple of years, we probably have greater insights into protection mechanisms with this pathogen, SARS-CoV-2, as compared to many other pathogens that we've worked for much longer using non-human primates. And finally, we learned about the consequential differences upon experimental infection in mice versus non-human primates in the context of malaria, but this is true for tuberculosis and probably many other complex infectious diseases, and how studying these in mice prevents the full complement of the immune system to be unraveled. And that is so important to study in non-human primates. And then I think it is also clear from all three of the talks that the expertise of the primate centers is so valuable. The fact that a new faculty member can come into one of these primate resources and have veterinarians, surgeons, pathologists that are just skilled enough to help with the development of these models. I'll just start with my own example. When I started at Tulane now 16 years ago, the veterinarians led by the group, led by Skip Baum and the pathologist there, Pete Didier, were really instrumental in teaching me how to infect monkeys with tuberculosis and do work with them. And this happens at every other primate center. And many of our speakers alluded to the various skills of these veterinarians and pathologists. And I think that is very important, that skill set and that expertise, that combined expertise of the primate centers is very important to acknowledge in these talks as well. David, Dr. Connor, there is a question for you. You mentioned administering immunoglobulins at different times post-Zika infection and that this displayed different disease outcomes. Why do you think this is? And specifically, if you could expand your thoughts on the miscarriage outcome.
Sure. So it was actually the timing of the infection that led to the difference in miscarriage. And we're still putting this together, but we're now at something like 10 out of 13 macaques that are challenged with an African lineage Zika early in the first trimester end up with miscarriage within a pretty consistent time window. So it's pretty reproducible. And I think that while our initial work started with hyperimmunoglobulin back in 2016, the success of monoclonals, especially with respect to COVID, shows that they really do have a place in therapeutics and can be administered at scale. So I think our future work is going to build on what my colleague and friend Sally Permar talked about yesterday when she gave one of these talks and mentioned some of the work that Matthias and others have done with therapeutic IgMs and some other innovations in monoclonals. So trying to see whether we can use some of those monoclonals to interrupt that fetal demise phenotype that we see early on. So develop the model and then figure out how to tease it apart to figure out why it's happening, and then figure out what sort of interventions can prevent it from happening.
Thank you so much. Outstanding research and a lot more of production and productivity and progress to come. Dr. Hartigan O'Connor, there's a question for you as well. How were the responses in CD8 and NK cells with S3?
Thank you, Deepak. Sorry that I can't seem to activate my video. I don't know, there's something wrong with my Zoom, but the CD8 responses, I would say, are underwhelming when you've been, working with, GAG and other intracellular proteins, and they're sporadic as CD8 positive T cell responses tend to be. So I would say that basically this approach of T cell targeting didn't have any obvious beneficial impact on the CD8 positive T cell response.
Thank you. And do you think there is some possibility that B cells, and now we can see you, Dennis.
Oh, good.
Do you think that there's proper crosstalk between B cells and T cells, and that B cells provide help in this instance for T cell activation?
I do. I think that's the most likely source of the augmentation of TFH responses is somehow coming from the B cells themselves, but they don't seem to be having that impact on CD8 responses.
Thank you, Dr. Murphy. There's a question for you as well, but it's from our co-panelist, Dr. Hartigan O'Connor. So Dennis, do you want to go ahead and ask that question?
Yeah, I was wondering if these cells that are known to play such an important role in controlling malaria that traffic to the liver are IL-15 dependent, because there's been a lot of information about IL-15 dependence in responses to rhesus cytomegalovirus vectors and to CMV more generally?
Yeah, so we haven't done that work, but Bill Heath's group in Australia has looked a lot using B6 mice. And in their model, I think they definitely found there was IL-15 dependence. The way they did it was they adoptively transferred in like OT1 T cells into regular B6 and IL-15 knockouts and found that they couldn't recreate the desired TRM phenotype in the IL-15 knockouts. We work in BALB/c mice predominantly, so we don't have quite as many knockout options. So we've looked to them for that finding. And it's complicated, but IL-15 is definitely part of the story.
Interesting. Thanks.
Thank you both. There's another question for you, Dr. Murphy. How different are pigtail macaques and rhesus genetically?
Well, that is a question someone on these attendees or panelists could probably answer much better than myself. I really can't tell you. What I can say is that on the parasite side, the host range for P. nolsi in Southeast Asia overlaps with that of pigtailed macaques, and it is unusual then for rhesus to get this in the wild. That was one of the inspirations for us to think the phenotype might be not as pathogenic, but we're kind of in early days of this, and that's certainly an interesting question. There's a lot of human host genetics that guide differences in susceptibility to malaria. In fact, people think that malaria may be one of the greatest genetic selective pressures on the human genome, at least in terms of infections. And looking at that with the same view toward rhesus and pigtails would certainly be interesting.
And I agree with you. And maybe rhesus and pigtails and cynos as well. There are stories that maybe David can tell us about how genetic differences between cynomolgus and rhesus actually end up informing us as to how they deal with tuberculosis, for example. Is quite
Sure.
interesting.
Well, I mean, I think this is a great example of how refinement through improved genomics is going to be the story of the next couple of decades of non-human primate research. We're always going to be constrained by the small numbers of animals involved in these studies. And we're always going to be working with animals that are have a natural biogeography throughout Southeast Asia, as well as the island of Mauritius. And we need to be able to understand what the impact of species selection, and then within a species, geographic selection has. And as we move more towards personalized medicine in people, I think you're going to see that non-human primates are going to be right at the forefront, because the marginal cost of generating a macaque exome or a macaque whole genome sequence or a macaque transcriptome is relatively low compared to the resources required for these sorts of experiments. And so we're going to want to know everything we can about these animals, even if we're not in a great position to interpret it right away. It might take quite a while for us to be able to decipher what the data means. But the exciting thing is that the technology is now there, that we can be collecting that data and starting to tease apart those questions.
Thank you, David. So definitely to powers that be a plug for more genomics expertise and more genomics support for all the primate centers is absolutely required. It looks like that there are no other additional questions. If that is the case, David, if I may ask you, and you briefly talked about it, but at the time of the beginning of the pandemic, you had also worked really hard along with your group. And I think your director, John Levine, mentioned this and shared some pictures at that point of time where you were literally working out of your van, and you showed some of those pictures to develop novel diagnostics for SARS-CoV-2. And then these were also applied in monkeys. And where is that story now?
Yeah, so early on, we took advantage of the knowledge that we had from doing SIV PCRs and SIV molecular diagnostic assays, and we pivoted to providing supply chain independent LAMP testing. Some of this was also done in California and around the country, and we deployed a bunch of it out of a minivan here, which was really important for the primate center because when testing was unavailable, primate centers are 24/7 operations. It was really critical that we try to protect the workforce. And so we were able to use that as a Band-Aid until higher quality antigen tests became available later in 2020. And then we were able to work to get a CLIA waiver for our group so that we could professionally administer BNAX Now antigen tests. We got 5,000 of them from the Assistant Surgeon General. And so we were able to help provide testing for some of the workforce at the Primate Center at a time when antigen tests had not yet become very widely accepted. And so, that sort of cutting edge translation between the Primate Center work, which is my background, and then bringing it out to the community really had a place there. And then more recently, we've been doing the exact same thing with air sampling, this idea that we can suck viruses out of the air, detect what's in an air sample. We have these running in schools around Madison, Wisconsin, which is where I am, as well as homeless shelters and a variety of other settings. We also have one at the Primate Center that Buddy Capuano, our attending vet, uses to keep an eye on the health of the air in the Primate Center to make sure that we can try to intercept any sort of respiratory pathogen outbreak before it becomes significant.
So that is such a good example of how the Primate Center's expertise is really not just informing basic science in non-human, using non-human primates, but actually goes far beyond that and has a strong translational impact. I think there are no more questions, in which case we can end the session. I want to thank all of my co-panelists and the speakers today for wonderful talks. I know that this is not easy to do, in terms of technology. And I know that your talks took a lot of time to prepare. So thank you all. And David, in your case, we wish you the best for your upcoming surgery. Thank you all. Goodbye.